All right, now we're going to go look a little deeper into bacteria. We're going to go over what endospores are and the different uses of bacteria in our world and how we as humans interact with them and what their job is. First, endospores. Endospores are pretty simple. Basically, whenever the environment's unfavorable for a bacteria, it's going to want to protect itself from that environment. So it forms a thick wall around all of its genetic material, because that's the important stuff. That's the code for what to do. If a ribosome or cytoplasm or cell wall gets kind of damaged, it doesn't matter as long as it has instructions on how to repair it. So it is going to encase the genetic material in what's called an endospore. Basically, when environment is unfavorable, this is going to form and the cell is going to go dormant during this process. When the genetic material is encased, there is no way for the cell to actually use it and start making proteins, so the cell is going to be inactive, when this is going to be a dormant stage. Again, it's used for protection when things are unfavorable. Now let's look at the role of bacteria in our world. So the role of bacteria has 5 different categories that we're breaking it down into, and the next slides are going to illustrate each of them. So that is producing oxygen, food production, environmental recycling, and environmental cleanup, as well as health and medicine. So first, looking at oxygen producing. So this is, again, our formula for photosynthesis. So make sure you are looking at this a lot. Make sure you are memorizing this. It will be needed on this test and further tests to go. Last chapter we looked at the fact that early life probably did not use oxygen because there was no oxygen in early Earth. So where did all this oxygen come from? Well, scientists are believing that the first oxygen on our Earth was actually the waste product from these microscopic single cellular organisms we call bacteria today. So as it goes through the photosynthesis process to make its own food, we're going to take that sugar or that carbon dioxide that's plentiful in our air, add it with the water that we are currently surrounded by, because again, scientists are going to say that living things arise from water and then move to land. Through the process of photosynthesis, we are going to get glucose, again, which is sugar and food, and we're going to get oxygen, which is our waste product. So all these bacteria are producing a waste of oxygen and throwing it into our air, and now our atmosphere is being filled up more and more with this oxygen. Next use for bacteria is food production. You probably eat foods that are somehow involved with bacteria in some processes. These foods include, like the list at the side here, cheese, sauerkraut, pickles, apple cider. The fermentation process of apple cider is through a bacteria. Buttermilk, yogurt, and sour cream. We also looked at last chapter a man called Louis Pasteur, and he actually devised our pasteurization process, very similar to his experiment with boiling broth. So again, for example, let's take our milk products. If we want to pasteurize our milk, what we're going to do is we're going to take that raw milk, we're going to heat it up hot enough to kill the harmful bacteria but keep the taste. So there's a specific temperature we are looking for depending on what our product is. Once it heats up, kills all that bacteria, it's then going to be cooled down and shelved, and this is called the pasteurization process. Now that all that bacteria is gone, and we know that spontaneous generation is not a thing, that we can then seal that up and it will have a longer shelf life. This pasteurization process, again, is not going to kill everything due to the fact that milk is still going to spoil, but it will get the majority of the harmful bacteria out of there, and it'll make your food products last longer. It'll give them a longer shelf life. So that's a very important process there. Next is environmental recycling. This involves 2 categories. One is decomposers, which I am assuming we are familiar with the process of decomposition. So basically when our organisms die, we need to break down their bodies, or we just have a whole bunch of bodies laying around what's just taking up space. So we do have organisms that whose job is to decompose these dead things and turn back the nutrients. So bacteria are one of those organisms that can be decomposers. However, they are also nitrogen recyclers, which is actually a lot more interesting. Um, living things, living organisms need nitrogen. However, nitrogen is hard to come by, and you're probably thinking, well, our air is actually 80% nitrogen. How come they don't just use that nitrogen? Well, that's because that's diatomic nitrogen. That's nitrogen bonded to itself, like the graphic shows here. And with nitrogen bonded by itself, nitrogen does have a triple bond. These three lines between our two ends over here represent a triple bond. That's extremely hard to break, and therefore most of the nitrogen in our air we can't use. So we got to find some way to convert it to nitrogen we can use. Lucky for us, bacteria can do that. Bacteria can actually break that triple bond and therefore provide nitrogen for plants to grow and nitrogen in return for us when we eat those plants. So that's what we call nitrogen recycling. Another aspect is environmental cleanup. The biggest thing you need to be concerned about this is that some bacteria love to decompose oils. So whenever we have an oil spill like the Exxon Valdez off Alaska or even the Gulf Coast, bacteria was introduced to the system in order to start breaking down the pollutants. So we don't want the oil floating around in our oceans or even in our soils when pipelines break. Bacteria is used to help clean that up. Our last one is health and medicine. There is currently bacteria residing in your body right now that help you with digestion, fighting diseases, and even producing vitamins that your body needs. Another way is through the production of insulin. Just like we were talking about with genetic therapy or gene therapy, with viruses, what scientists do, they will inject certain bacteria with a virus that is coded to make insulin. So this virus will take over the bacteria cell, and in return, that bacteria cell will stop doing what it's supposed to do and start producing the protein insulin instead. They put these in huge giant vats and eventually just let the proteins make all those, or let the bacteria make all the protein for insulin, and we then collect the insulin, which we can use for diabetes.